Hi there, everybody. My name is Michael Lay, and for the purpose of my UNE online physiology course, I'll be discussing the overall ins and outs of fat digestion today. So let me minimize my screen here so you guys can see the presentation. There we go. So I'm going to be running you guys through this presentation where I talk about the overall process of fat digestion within the body. And then I'll be discussing a clinical case scenario and help you guys answer questions related to it. So let's talk about that, shall we? So the overall goal of this presentation is to help you guys understand the why, the where, as well as the how of fat digestion. So I'll be answering those questions as we move along further. So lipids are actually one of the three main sources of energy that humans use. We use carbohydrates, we use proteins, and of course, we also use fat. Now, Americans actually consume over 50% of our daily calories through low quality carbohydrates and saturated fat, which is very bad. And more. I'll discuss this process more and why it's bad in the upcoming slides. Now, let's answer that question, where? So, where does fat digestion occur? Now, fat digestion begins in the oral cavity, where fat that we've ingested, like this egg, for example, interacts with lingual lipases that initiate the breakdown of these triglycerides. Now, what are triglycerides? Well, these are the main components of fats. So in the medical field, you'll hear the terms triglyceride, triacylglycerol, lipid, and fat all interchangeably. So I'll touch a little bit on that on the next slide. But let's focus on the where. So food is mechanically or physically broken down into a ball called food bolus. So it's covered in salivary amylase, lipases, and other digestive enzymes that help break down these molecules. The food ball of semi-digested carb, protein, and fat then travels via peristalsis. So if you guys are looking at my hands, you'll notice that it's like a wave-like movement. So it travels via peristalsis through the esophagus and into the stomach. And peristalsis is, of course, innervated by our autonomic nervous system. So the stomach, very important, is where emulsification occurs. So this is a process that increases the surface area of fats, making them more susceptible to lipid breakdown via digestive enzymes. So these enzymes I mentioned, primarily lipases, then break these longer chain fats into smaller, more digestible, get it? Digestible? Bits. So remember that word bits. So breakdown of lipid into these bits is continuous. It starts in the stomach, but mostly occurs and ends in the small intestine. Now, again, these bits, they may eventually become diglycerides or even small, medium, or long chain fatty acids, which have very important biochemical significance, particularly in the TCA cycle. But I'm not going to bore you guys with biochemistry. So let's talk more about the enzymes and how they make this all happen. So let's answer the how, right? So you'll notice an etymological uh, pattern on the slide. You'll see, repeats lipase, lipase, lipase. Now remember that. We know that lipid is another term for fat. And so therefore, we can ascertain that the enzyme lipase is the primary enzyme responsible for lipid breakdown. Now, in the mouth, we've got lingual enzymes. In the stomach, we've got gastric enzymes, um, lingual lipases, gastric lipases. And then in the pancreas, of course, that secretes pancreatic lipase. Now, let's talk about bile. Bile is a very important key player in the mechanism of lipid digestion. So bile is actually manufactured here in the liver, as you can see here. So it's manufactured in the liver and it's stored in the gallbladder just below it. So Bile then travels from here, the gallbladder, or gallbladder, yep, down through the common bile duct to the pancreas. And from the pancreas, via the sphincter of Oddi, it'll move into the duodenum and finally up the pyloric sphincter and into the pylorus or the bottom half of the stomach. So bile initiates, bile again initiates the emulsification of fat. So it makes it easier for these lipases to break down. Now, as I mentioned before, this is a continuous process actually. So it begins lightly in the stomach, but then the vast majority of lipid breakdown occurs in the small intestine. But it doesn't end there, right? The journey of lipids actually doesn't end in the small intestine. Of course, our body needs to utilize this lipid somehow, right? So let's talk about cholesterol. Now, cholesterol is made of fat and can be transported by, well, you guessed it, other fats. We'll call these chylomicrons. Now, chylomicrons are large triglyceride-rich lipoproteins, so lipid and proteins, lipoproteins, produced in enterocytes, or cells of our small intestine. These are produced from dietary fat. Now, as you can see here, chylomicrons can then undergo maturation and conformational changes that may convert it to VLDL, LDL, IDLs, and HDLs, which are various density lipoproteins. And of course, these have clinical significance. Now, HDLs are good, and you'll notice that they are smaller. So HDLs transport cholesterol across your tissues to the cells that need it. And LDLs here, so here's a VLDL, very low density lipoprotein, and LDL right here. So VLDLs and LDLs are actually very bad. So if these huge, relatively speaking, um, density lipoproteins circulate too much in the bloodstream, they can actually clog your arteries by forming plaques. They can also sort of connect to blood glucose. So this may cause damage to the arteries and can even lead to nerve damage as well. And I'd like you guys to remember that detail. Now, let's talk a little bit about biochemistry. So I just wanted to stress how important fat is to our body, right? So a final application of fats in your body is HDL transport of lipid to your cells. Now, this is callback actually to Unit 1, where we learned about how cholesterol transport across the cellular membrane is receptor mediated. We learned that cholesterol can contribute to various cell membrane functions, influencing its fluidity and its structure. Now, fatty acid transport is also receptor mediated. And see, triglycerides become eventually are broken down by lipases, they become fatty acids. And then these fatty acids bind to a receptor on the cellular membrane called FFAR, or free fatty acid receptor. I know it's a very creative name for the receptor. And of course, this also directly contributes to the Krebs cycle. And why is the Krebs cycle important? Well, the Krebs cycle helps our cells produce energy, or ATP, as well as break down our proteins and then disperse them as amino acids throughout the rest of the body. So the Krebs cycle is very important to our energy production. Now, just to recap, at the beginning, I asked you guys to recall the where. And so in answering that question, I talked about the anatomy of the fat digestive process. And then I asked you guys to ask why. And so in why, I want to talk about or why rather, it's important. And I then discussed the various mechanisms of cholesterol and triglyceride synthesis or triglyceride breakdown into fatty acids. And finally, I asked you guys to ask yourselves the how. And the how is the enzymatic processes, the enzymes that drive the process of lipid digestion. Alrighty, so that covers the presentation of lipid digestion. Now, I'd like you guys to read over a case study with me. Let's talk about this case study. So, a 40-year-old male patient with no significant familial medical history enters the clinic complaining of sudden severe unilateral pain in the hip and lower back. He also reports numbness in his lower extremities and a consistent burning pain in his left foot. So, blood workup with lipid panel reveals an HbA1c count of 9%, 182 milligrams per deciliter LDL count, 72 mg over dl HDLs, and 200 mg over dl triglyceride. Now, the patient is mildly obese, and the nurses report a putrid odor in the patient room. So, sometime later, the patient's left limb is partially amputated, as you can see here in the image. Now, what happened here? Let's answer these questions now. So I'm going to be doing this on a worksheet that you guys are able to see now. And let's answer these questions, shall we? So, so what happened to the patient? So this patient is suffering from diabetic neuropathy. So I'll just actually briefly put it right here. Diabetic neuropathy. This occurs from damage to peripheral nerves by high concentrations of blood glucose, which is related to those low density lipoproteins, as I've mentioned before. So, high blood glucose is associated with high blood pressure, high blood cholesterol, and of course, the disease that we are going to talk about in the next question. So, which disease does this patient likely have? And how can you determine the type of disease given the clinical presentation? Now, this patient likely has type 2 diabetes. So now, this is indicated by the lipid panel. So you'll notice that he has a very high, very high LDL count, which is bad, rather low high density lipoprotein count, high glucose percentage, and a very high triglyceride count. Now, this is obviously diabetes, but why do we know it's type 2, right? So this is actually determined because it was specifically stated that there is no significant familial medical history of illness. So we know that type 1 is inherited usually, and type 2 is more caused because of lifestyle factors and isn't necessarily correlated to genetics. And what specific lipid mechanisms are disrupted? And how did this lead to the patient's partial leg amputation? So type 2 diabetes is a result of suppressed insulin signaling in the pancreas. This leads to central obesity, overproduction of VLDLs and LDLs, as you can see through the lipid panel, and of course, high blood pressure and sugar. High blood sugar you can see in the blood panel as well. So, as discussed in the PowerPoint, LDLs in the bloodstream can combine with circulating glucose in the arteries, causing damage to both arteries and nerves. So these peripheral nerves are damaged by this accumulation of stuff in the circulating arteries. And of course that is exhibited by the artery damage and then vein and then eventually nerve damage leading to um, this leading to the patient's clinical symptoms as seen in the case. So that concludes my video on digestive system, fat digestion. And I began with a PowerPoint, talked about the ins and the outs of lipid digestion, and then I also introduced a case study for his students to look at as they further understand the processes involved in lipid digestion. Alrighty, everybody. Well, thank you for joining me for this presentation, and I hope you guys have a lovely day. This is Michael A. signing out. Bye bye now.